All right, so this is the front part of chapter 13. This really deals with genes and molecular biology. I'm going to describe the structure and the function of DNA. Excuse me. I'll describe the genetic code and how cells make protein. So this is kind of really following up on Mendel. Mendel had identified these factors of inheritance that explained traits that could appear and disappear. He had identified dominant and recessive factors, and that individuals receive factors for each trait from each parent. But uh, we didn't really know what Mendel's inherited factors were. We had no idea, but we knew they had to be in cells. And we didn't really even know how those factors produced traits. Um, what were the traits? What were the factors? It was all unsure. Well, that guy right there, he was one of the first guys to really kind of connect things to chemicals in cells. And he worked with human diseases. His name was Archibald Garrett, and he was active right around 1900. And he recognized that one particular disease was transmitted just like Mendel's inherited factors. It was a recessive allele or recessive gene, and it caused a kidney disease called alkaptonuria. Um, and these people, their urine was black because there was an amino acid that they couldn't break down. And so when they excreted it, their urine was black. And he found that these people were missing a particular enzyme. And so that connected Mendel's inherited factors to enzymes, and enzymes were proteins. And so we knew, okay, the inherited factor results in the production of a protein. But then we really weren't sure what was the inherited factor until those guys on top there, that's James Watson and Sir Francis Crick, they proposed a model for the structure of DNA. And there had been a lot of information building that up over time that the inherited factors had to be in the nucleus, and they were probably related to the chromosomes. We knew that if you transferred a nucleus from one cell to another, that that cell began to look like the cell that the nucleus came from, and that the nucleus could direct inherited factors to make traits. We also knew from karyotypes that there were chromosomes in cells, these large conglomerates that contained lots of DNA, but also some protein. Later on, we discovered that we could transfer inherited factors when we transferred DNA. This happened with bacteria and viruses. And the big breakthrough came when this woman in the center there, uh, she took pictures of DNA, and it showed the structure of DNA as being a double helix. These pictures were X-rays of DNA crystals. And from that, they could tell that there were two chains and that they were a certain distance apart. And so this kind of really nailed it that the uh, you know, the inherited factors were in the nucleus, they were in the chromosomes, and they were the DNA that was in the chromosomes. And uh, once we knew the structure, we could understand, okay, DNA is probably the genetic material. And that was because DNA had to be able to store information, be able to be copied exactly, very accurately for future cells. It had to be able to be used to express the traits, but it had to occasionally change the information and mutate. And so once we knew the structure of DNA, we kind of knew that, okay, it can do all these things. So DNA is made up of, it's a polymer made up of a large number of subunits. These subunits are called nucleotides, and each one contains phosphates, sugars, and a base. And the phosphate here is shown in yellow. The base is the blue, or the sugar is the blue-green part, and the base is shown in purple. And in DNA, the sugar is always deoxyribose, so it has an H right in this spot, that lower down. And then the bases are adenine, guanine, thymine and cytosine. We had known about the structure of nucleotides, but we didn't know how it worked until we had saw something about the structure as having a double helix with side chains of sugar phosphates forming a backbone, but then a sequence of bases kind of tucked in. And these bases held the two DNA strands together by forming complementary bonds between thymine and adenine, where they were held together by two hydrogen bonds, and then cytosine and guanine, which were held together by three. And this was the only pairing that was allowed in DNA, but it meant since these were complementary, if you knew one half of the chain, you knew the other half. And so when they realized that there could be any sequence here at all, that the sequence, um, it just mattered that they were complementary, then they understood that DNA could be a code for making protein, that any three bases in the DNA could be converted to one of 20 amino acids. And it turned out that this was the same in all cells using the same genetic code and the same process for converting a DNA sequence into a sequence of amino acids in a protein. And this involved a couple enzymes and a couple molecules. And this structure also explained kind of nicely how why DNA could be easily copied. So you could copy all the genetic information by just separating the two strands, and each strand would serve as a template then for making a new strand. That an enzyme could just add complementary bases, and so you could copy DNA very, very easily by just separating the strands. And at the same time, this wouldn't affect the code. And so the code was three bases used to make an amino acid. And so the intermediate step is making a messenger RNA, which substitutes uracil for thymine. And these are different three base sequences code for different amino acids. And so here they're shown right there, all the ones. You don't have to memorize this. It's just we know the genetic code and it's the same in all cells. We also then eventually figured out that there were two steps in making a protein. One was called transcription, where an enzyme could add complementary bases to a growing RNA chain that was made from a DNA template. And then the second was translation, where that RNA uh, template could then be used to make uh, a DNA molecule, or not a DNA, a protein molecule, which was a chain of amino acids. So the first step required one enzyme called RNA polymerase, shown up top here as green, that would open the DNA strand and start adding complementary RNA bases based on the DNA template. This would cause the production of a messenger RNA or mRNA. And then that mRNA would go into the cytoplasm where a ribosome would assemble on it. And the ribosome had spots for transfer RNAs to bring amino acids. And the ribosome would link these amino acids together, and then it would get rid of uh, the empty transfer RNA molecule and then move just a little farther down to reach the next codon. So once we found the structure of DNA, it became pretty obvious how it had to work. And this is the basis for molecular genetics and molecular biology, that this genetic code is pretty universal. It allows us to move genes from one organism to another, and it suggests all organisms share a lot of common ancestry. Of course, this is made possible genetic engineering. Here we've got two pigs. One has had a gene for a fluorescent molecule inserted into its DNA, and that molecule, that fluorescent molecule, was actually first found in jellyfish. But if you insert that DNA into the embryo, the pig makes it. Okay, and we found that this code is practically universal. There's only minor differences in mitochondria and chloroplasts. And so we'll go over some more details. I'll give you some examples of how this works, talk about some of the things that they do next time. Uh, that they can do with molecular biology and and some applications of this technology. All right.